Revolutionary biology discovery combines DNA and RNA to fight cancer. By Free University of Brussels, February 2nd, 2025. Recent research reveals that DNA and RNA epigenetics, previously studied independent systems, actually function as a complementary regulatory network. This dual mechanism enables precise gene activation, critical for cellular development and function. Published in Cell, the study uncovers a novel mode of gene regulation and its role in diseases like cancer, paving the way for personalized therapies using epigenetic drugs that target both DNA and RNA.
Epigenetic. Epigenetic. Epigenetic.
A study reveals DNA and RNA epigenetics work together as a complementary system for precise gene regulation, crucial for cell function and development, with implications for advanced cancer therapies. Our genes carry the complete set of instructions necessary for our body to function, but their expression must be carefully regulated to ensure that each cell fulfills its specific role efficiently. This is where DNA and RNA epigenetics come into play, a collection of mechanisms that act like markers on genes, controlling their activity without altering the underlying DNA or RNA sequences. Traditionally, DNA and RNA epigenetics have been studied as separate systems. These two mechanisms were thought to operate independently, each contributing to different stages of the gene regulation process.
RNA. RNA. RNA.
In a publication in the journal Cell, researchers led by Francois Fuks, the Laboratory of Cancer Epigenetics, ULB Faculty of Medicine, ULB Cancer Research Center, and Jules Bordet Institute HUB reveal that, in fact, DNA and RNA epigenetics could be more interconnected than previously thought. The researchers have discovered that they form a complementary regulation system in which DNA epigenetics organizes the available genes and RNA epigenetics dynamically adjusts their use. How dual epigenetic markers work. In concrete terms, the study demonstrates that when these two markers are added jointly to a gene, they enable a more effective activation of that gene. On the other hand, if one of these processes is not working correctly, the gene's activity diminishes. Francois Fuks and his colleagues have shown that this mechanism is particularly important in key stages like cells development or their specialization into different types, for example, in embryonic stem cells. This combination offers incredibly precise regulation of gene activity, essential to the development of organisms and the harmonious functioning of cells. Implications for biology and medicine. Published on January 17th in the journal Cell, this fundamental breakthrough sheds light on a completely new mode of gene control, opening up unprecedented perspectives in biology. It helps us to gain a better understanding of how our cells work and how disruptions to these mechanisms can cause diseases like cancer. This discovery could also lead to advances in cancer treatments. Making use of this complementary regulation system raises the prospect of developing therapies based on epigenetic drugs that target DNA and RNA at the same time. The scientists hope to be able to develop more precise and personalized treatments capable of targeting these regulation mechanisms to restore balance to diseased cells in cancer patients. Prof. Fuks' team is already carrying out research connected directly to the work published in Cell. These ongoing studies aim to demonstrate the clinical usefulness of their discovery by exploring the potential of epigenetic therapies acting on DNA and RNA. Reference: fine-tuning of gene expression through the METL3-METH14 DNA one-axis controls ESC differentiation by Giuseppe Quarto, Andrea Ligrei, Martin Bizet, Audrey Penning, Irina Primac, Frederic Murigier, Liliana Garcia Martinez, Rodrigo L. Borges, Qingzhen Gao, Pradeep K.R. Singaram, Emily Cologne, Butra Hasabi, Celine Hubert, Adele Herpol, Pascal Putmans, Frederic Mies, Jerome Martin, Louis van der Linden, Gaurav Dube, Pankaj Kumar, Romuald Soin, Abhay Kumar, Anurag Misra, Jilan, Morgan Pak, Yogesh K. Gupta, Arnaud Blom, Pierre Close, Pierre-Olivier Esteve, Elizabeth A. Kane, Kristen M. Riching, Cyril Guidan, Danette L. Daniels, Sriharsa Pradhan, Raman Shikhatar, Yale David, Louis Maury, Jana Jeschke, Rachel Deplis, Eveline Collignon, and Francois Fuks, January 17, 2025, Cell, DOI 10.1016J Cell, 2024 12009.